good practices in shrimp aquaculture. Okay, so so there are three main reasons why we want to have a healthier shrimp. So in the context of the shrimp farmers, we want to produce, or fish farmers want to produce healthier shrimp so that they will have good production as well as for profit. And that was clearly shown to you in the previous talk. The second issue why we need to have a healthy shrimp is that we want to make sure that our resources would be sustainable. And that's also the role of probiotics, where in terms of bioaugmentation, we wanted to have a clean pond bottom, as well as we wanted to have our waters to be clean from these different kinds of waste materials. And the third issue of why we need, or why we are advocating for healthy shrimp, is that we wanted also to make sure that our consumers are assured of safe and high quality product. So these are the different three issues why we need to have a healthy shrimp. So my presentation for this afternoon is divided into four parts. So we have an introduction on probiotics. So I think this has been given by the previous two talks, where they have presented some background on probiotics. And then I'll discuss about probiotics and their effects on shrimp. And then I'm going to give you a brief overview of the shrimp immune system. And lastly, I will discuss about gut health and microbiota. Now, sustainability of any aquaculture operation, and that would include shrimp farming, these are met with different issues. And then we also have the issue on aquaculture mediated species invasion, and also the problems with the abuse of chemicals that we are using in our aquaculture facility. And lastly, the issue on diseases, particularly the presence of infectious diseases. And no need to elaborate further, because these issue on the presence of infectious diseases have been underscored in the previous talks. And here we have known that infectious diseases in shrimp farming, these are caused by a number of disease-causing agents, such as parasites. You have the bacteria, and you have the viruses. So example of a bacterial disease would be vibriosis, and this is caused by various species of Vibrio. You have now, before it used to be luminous Vibrio, then you have the append. And these are caused by different species of Vibrio. And also you have the white spot syndrome, which is an example of a viral disease. And this one is caused by the dreaded white spot syndrome virus. So based from experience, we have known that uh, in the past decade, our country has suffered drastic losses in terms of shrimp production because of the presence of these infectious diseases. So given the severity and the impacts of these infectious diseases in shrimp farming, particularly those of bacterial origin, so shrimp farmers normally in the past, they have resorted to using antibiotics. However, we now know that the use, or the abuse of antibiotics resulted in negative consequences, particularly on the indiscriminate use of these chemicals in shrimp farming. As such, would the shrimp farmers, would they have an alternative to the use of antibiotics to treat bacterial diseases in shrimp? So they have been debating, or whether do we really have an alternative for that. to the FAO, it has recommended three areas that need further research in the prevention and control of these various infectious diseases, not only in fish, but also in shrimp. And these would include the development of your vaccines. You have the development or application of immunostimulants, and of course, the development of probiotics. So the role of probiotics will now be discussed further in this talk. This has also been given to you by the previous talk on the interaction of probiotics in water quality, as well as the role of probiotics in the bioaugmentation in shrimp ponds. So the term probiotics came from the Latin word pro, meaning for, and the Greek word bios, that means life. So simply stated, probiotics are good bacteria. Now, our traditional definition of a probiotics, and this is in the context of animal nutrition. So we define probiotics as a live microbial feed supplement which beneficially affects the host animal. So that is in the context of human or animal nutrition. And a proof of that is why we are drinking Yakult, because we drink Yakult because it contains probiotics which act as a feed supplement. Now, so the preceding definition of probiotics is based on the context of animal and human nutrition. So what about in the context of aquaculture? Now, based from published studies on the use of probiotics in aquaculture, we have defined probiotics then as live, they could be dead, or they can be a component of the bacteria that act uh, under different modes of action in conferring beneficial effects to the host or to its environment. So in the previous talk, they mentioned about bioaugmentation, where probiotics have been added to the pond system, and that is affecting the environment of the shrimp. In the same manner, probiotics have been added in the feed, and that would affect the host. So in the context of aquaculture, probiotics have been defined as anything, or it could be alive, it could be a dead, or it can be a component of the bacteria that had different modes of action and will have beneficial effects either to the host or to its environment. So from a diverse population of bacteria, how then do we select probiotic candidates? So we have developed different selection criteria. So the first of these first criterion is the appropriateness. So under this category, it includes the accurate taxonomic identification of your probiotic candidate. And also, you make sure that your probiotic candidate is non-toxic and non-pathogenic. So it has achieved a GRAS status, or it's known as a generally regarded as safe status. So that's the first criterion of selecting a probiotic candidate. The second one is technological suitability. Under this criterion, we consider factors such as the candidate must be amenable to production and storage. They must exhibit high viability, as well as they must be genetically stable. So I've discussed appropriateness, technological suitability, and the next one would be competitiveness. In this criterion, we consider factors such as the ability of that candidate to resist the action of bile, to resist the action of acid, as well as its ability to adhere and colonize the target tissues of the host. And the last criterion would be the performance and functionality. Under this criterion, we should be able to document a health benefit of that particular candidate, of that particular probiotic candidate to the host, as well as we need to determine whether it has an antagonistic activity toward the pathogenic bacteria. So it's also under this performance and functionality criterion where we also determine whether the probiotic candidates or the probiotics that we are using have the ability to produce bioactive compounds. In this case, if they produce some digestive enzymes, then we are assessing the performance and functionality of that particular probiotic candidate. Now, this slide would show you how probiotics are being developed. So probiotic candidates can be obtained from either the host itself or non-host origin. After you have isolated some of these candidate microorganisms, these will be followed by in vitro studies. And during the in vitro studies, we determine the properties as well as the presence of inhibitory substances found in these candidate probiotics. Now, after you have done the in vitro studies, you now have a candidate probiotics. So please don't have the impression that after you did the in vitro work, you already have the probiotics. So it's wrong to say that you already have a probionts. You only have a candidate probiotics after you have undergone the in vitro studies. So after you did the in vitro studies, you now proceed to the in vivo studies where you check the pathogenicity of your candidate as well as you determine the beneficial effects to the host. Now, after you did all this in vivo work, you are now ready to register as well as prepare the papers regarding compliance to laws. And you can now market your candidate organism as a probiont or as a probiotic. So this is how normally we screen and develop probiotics, not only in aquaculture, but in other industries. Now, because of the growing popularity of probiotics in aquaculture, then you might ask the question whether the use of this is actually new or is it something that has been used in the past. And just for the information of everyone, according to this website, sciencedirect.com, as of November 2020, this month, there are more than 2,000 publications that are available where it highlights the use of probiotics in aquaculture. So that means that it has been used in the past and it is indeed popular. Now, if you include Philippines in the search criterion, you will get at least 100 publications on the use of probiotics. So that means that here in the Philippines, it's also gaining popularity, and it's not even a new concept. And in the EU or in the European Union, there is already an approved probiotic mixture for aquaculture, and this is marketed as Back to Cell. So Back to Cell is a probiotic that is coming from a lactic acid bacteria, and that is from Pediococcus acidilactici. So that's the strain or the genus of the bacterium that comprises the Back2Cell. So Back2Cell is a probiotic product, and that is considered as generally regarded as safe product, and they are categorized as a feed additive. So they are marketed as a feed additive in the EU. So from these studies on the use of probiotics in aquaculture, so around 90 works use either lactobacillus and bacillus. So even if you check some of these commercial probiotic products that we have, normally it would contain bacillus or a mixture of different bacilli and lactobacillus. Moreover, issues on whether these probionts are able to colonize the gut and their modes of action on the host are not clearly elucidated. So some of these probiotics, it gives you an information that it has beneficial effects on the host, or it produces digestive enzymes. But some of these products, they have not really elucidated uh, whether they are able to colonize the gut or whether they have produced the necessary digestive enzymes. So it's some of the issues that need to be addressed by research. Now, if we use probiotics in shrimp farming through direct application to the water or through dietary administration via the feeds, so this slide will show you the possible effects on the host. So what are these effects? They could modulate the host microbiota, and they modulate the host microbiota by inhibiting pathogens, or they could colonize the digestive tract. And also, once they colonize the digestive tract, they can also produce enzymes that would increase the production or the increased digestibility and nutrient utilization of the feeds. Therefore, there is improvement of the growth. Another effect would be they are capable of modulating the host immunity here. Therefore, they could stimulate both the cellular and the humoral defenses. And also, probiotics can directly inhibit the pathogen by producing inhibitory metabolites or inhibitory molecules. And also, they could affect the environment of the shrimp by influencing both the water and the soil quality. So these are the different modes of action that a particular probiotics could possibly affect. So we have mentioned that probiotics can stimulate. So one of the actions of probiotics is its ability to stimulate the immune responses of the shrimp. So unlike vertebrates, the shrimp immune system is mainly composed of the innate or your natural immune system. So it's unlike humans where we have both the innate and then the adaptive immune response. In shrimp and in crustaceans in general, they only have an innate or their natural immune system. And its innate immune system has a cellular response, which is facilitated by phagocytosis, nodule formation, and encapsulation. On the other hand, they will all the innate immune system of shrimp would have the humoral component. So humoral, or the what you call this, the liquid, the humor or the liquid part. And under the humoral immune responses, this would include the mechanisms that are involved in the prophenol oxidase or the propio system, the clotting system, as well as the antimicrobial system. So this slide would give you a much clearer illustration of the cellular immune response of the shrimp. So cellular immune responses in shrimp are carried out by the hemocytes, or these are the cells that are found in the hemolymph or the blood of the shrimp. So each type of hemocyte will have its specific function. So for example, you have a granulocyte and the hyaline cells. So these are hemocytes. So these granulocyte and hyaline cells, they are able to perform phagocytosis. That means they are eating or cellular eating. They're eating the pathogen that they come in contact with. On the other hand, the granulocytes, they are capable of nodule formation that will also form to engulf the pathogen. So each of these hemocyte will have their own specific function based on its morphology. Okay, and this one would be the humoral defense system. So in the humoral defense system, the cell come in contact with a pathogen. So here, these are the products of the pathogen. And once these would come in contact with the pathogen, it will activate the ProPO or the prophenoloxidase system. And once it's activated, these would be activated to the phenoloxidase and to the production of your melanin as well as your reactive oxygen species. If the phenoloxidase system is activated, leading to melanization, melanization is also implicated in the bacterial killing activity of the cells. And aside from the activation of the prophenyl oxidase system, it also activates the clotting system by a series of enzymes. Once the clotting system is activated, this would permit wound healing of the tissue or the cells at the site of the infection. And lastly, it also triggers the production of your antimicrobial peptides that would result in the lysis of the bacterial pathogen, as well as the production of your reactive species that will also have a bacterial killing effect. So if you have noticed, the humoral defense system or the humoral defense mechanisms of shrimp would always be focused on the antibacterial activity. So these are some of the identified immune-related genes in shrimp, and some of these are associated in the gut immune system as well. So here is an illustration of how probiotics might influence the immune system of the shrimp. So when probiotics come in contact with the shrimp hemocytes, it triggers an immune cascade by the production of the reactive oxygen species, which have antibacterial effects. It will also result in the proliferation of the hemocytes that will activate the prophenoloxidase system as well as phagocytosis. So both systems here will also have an effect towards a killing of the bacterial pathogen. And lastly, stimulation of trigger the transcription of the immune-related genes that will also be helpful towards providing protection of the host against this different pathogen. An influence on the shrimp immune system. Now, so how about in the shrimp gut? So what are the effects of the probiotics in the shrimp gut? So we have mentioned before, it was also shown in the previous talk, that the shrimp digestive system is divided into three regions. So you have the foregut, which is composed of your mouth, of the esophagus, and the stomach. Then the midgut of the shrimp is composed of the hepatopancreas, which is the site of absorption of the nutrients. And then you have the hindgut, which is composed of a chitin-lined tube or the intestines, as well as the rectum. And previous studies in shrimp have shown that, for example, in the tiger shrimp or in Pineus monodon, bacterial groups that were found in the intestines of post larvae and juveniles. And these bacterial groups are in the phyla of Bacteroides, Firmicutes, and Proteobacteria. Noteworthy to mention would be the dominance of Proteobacteria in the gut of host larvae and juvenile tiger shrimp is the presence of Proteobacteria, because Proteobacteria is a good source of endogenous probionts, or what you call as the host-derived probionts. At the same time, in white-legged shrimp or in Lithopineus venemei, a previous study also have shown that they were able to identify and isolate 64 bacterial strains that exhibit proteolytic activity, or bacteria that have the ability to digest proteins. And these bacterial strains were isolated from the stomach, hepatopancreas, and sequencing of these different strains, they found out that these belong mostly to the genera of Pseudo-Alteromonas and the Vibrio. And they have also discovered that the strains exhibited amylase, chitinase, esterase, and lipase activities, and these are can be considered as your digestive enzymes. So we now have a probiotics and the pathogen that they and how they interact in the gut. And I will just briefly show you the different models of how they interact in the gut of humans, fish, and shrimp. So we have a latest study in humans that showed that the gut microbiota. So we have previously known that the human gut, the microbiota in the human gut, are able to produce antibacterial antibiotics. However, this latest study has shown that the microbiota of the human gut also produce antiviral compounds, and these antiviral compounds are interferon response molecules. So this study is the first report that highlights such mechanism, because it was previously believed that the human gut microbiota can only influence the control of bacterial pathogens. So what about in fish? So in fish, we did experiments using host-derived probiotic candidates, and a bacterial pathogen that we have used is Aeromonas salmonicida. In here, we have shown, so we isolated the gut, cultured the different regions of the fish gut, and then incubated them with Aeromonas salmonicida. And from here, we have shown that this particular bacterium has the ability to colonize the different regions in the gut of the fish. So here, there was negligible adhesion in the foregut, but dominant adhesion in the other portions of the gut of the fish. Now, in the presence of a probiotic candidate, so we call it probiotic candidate A, it altered the dynamics of the pathogen. So in the case of candidate A, the mechanism of inhibition of the bacterium is through competition. Now, we use probiotic candidate B. And when we use probiotic candidate B, the mechanism of interference, or the mechanism of inhibition that we have noticed was exclusion. So from here, we have shown that each probiotic candidate will have different effect on how they would exclude or how would they compete with a potential bacterial pathogen. So this was one of the first reports of clearly elucidating the effect of a probiotic candidate in the gut of a fish. Unfortunately for shrimp, we haven't done that because it's quite difficult to develop a cell line, a primary cell line for the shrimp gut. So now in shrimp, we have this concept of a healthy gut, healthy shrimp through the use of probiotics. So how is this possible? So you have here a probiotics that would be incorporated in the feed and would be ingested during that would be ingested by the shrimp when you feed them with the feeds that have been coated with the probiotics. Now, once these are ingested, these are taken in by the shrimp, these probiotics or these probionts are met with biological barriers. So these biological barriers would include your stomach, such as the presence of a gastric meal. So the gastric meal would make sure that there would be mechanical shearing of the probiotic cells. And also it will, or your probiotic or your probionts would also face the enzymes produced in the hitopancreas as well as the enzymes that are produced in the intestines. So these three are barriers for the probiotic to infiltrate the tissue of the shrimp. Now, once the probiotic candidate or the probiotics would surpass these barriers, it will now travel to your digestive system. So if they are able to survive, the probiotic candidate must have the ability to colonize the gut, or as what the previous speaker has mentioned, it can uh, coat the entire lining of the gut, as well as it must produce the beneficial enzymes, as well as the byproducts that will aid in digestion. So if a particular probiotic would have these characteristics, then we can say that this particular shrimp now will have a healthy gut because it will have a dominance of these good bacteria, as well as these good bacteria would be the ones to produce the beneficial digestive enzymes that would aid in digestion, as well as to make the shrimp better assimilate the nutrients. Now you have here a better assimilation of the nutrients, as well as the presence of the dominance of these good bacteria. So what would then be the effect? So the effect would be an increased growth of the shrimp through better assimilation of nutrients, as well as because of the dominance of these probionts in the gut, it will inhibit the proliferation of bacterial or maybe viral pathogens, which I think needs further research. And once this is attained, then we have a healthy shrimp. So in effect, if you have a healthy shrimp, it all starts to having a healthy gut. So I have here a slide that shows how our shrimp industry will benefit from a strong partnership among the ancillary industries and the academe or the research institutions. So this is in line with probably probiotics development or any other um, uh, activity that will support our shrimp farming industry. So the direction can be both clockwise, that is from the shrimp farming industry to the ancillary industry to the academe and then back to the shrimp farming industry. Or it could be counterclockwise. So from the shrimp industry to the academe, then to the ancillary industry. So regardless of the direction, the shrimp farming industry starts with a need. And that need is channeled either to the ancillary industry or to the academe. And once the need is identified, it will trigger a series of events. And given this linked interaction among the stakeholders, it can only be successful if there is a provision of government support. So, in conclusion of this talk, the use of probiotics to attain a healthy gut, healthy shrimp, must satisfy the following requirements. It must have the ability to surpass the digestive barriers. These probiotic or probionts must colonize the gut